Now, how does knowing that Src is a tyrosine kinase help us understand signaling? Okay, so we know that the tyrosine kinase can go away and affect lots of other proteins, but what was also really important is that once you knew the gene sequence of Src, you knew where the tyrosine kinase domain is, you could then identify other genes in the cell that had tyrosine kinase domains. Okay, and it turned out that these receptor molecules in the plasma membrane, a lot of these receptor molecules also contained a tyrosine kinase domain. So the cell has this tyrosine kinase activity that's important in controlling signaling. Okay, so the epidermal growth factor, which is one of these key proteins that controls cell growth, it contains a domain that's similar to Src. So EGF is found on the surface and it stimulates growth through these mitogenic factors. Now, if epidermal growth factor gets mutated, it loses the ability to regulate and control itself. So it tends to be active driving growth when it shouldn't be active driving growth. So mutations in EGF as an oncoprotein drives growth, and we understand now that it has a tyrosine kinase domain that's phosphorylates of other things. And with this receptor, it has a domain on the outside that picks up the growth factor, but it has a domain on the inside that phosphorylates tyrosine kinase domains. So here we have the sequence of the Src protein that had been identified as being an important oncoprotein. When people then looked for this sequence in other genes, they identified a lot of these receptors, these tyrosine kinase receptors, that have a tyrosine kinase domain on the cytoplasmic domain of the protein. So this is the membrane here. This is a transmembrane protein. It picks up a growth factor on the outside of the cell, which turns on the tyrosine kinase inside the cell. And people identified this because of the homology to this Src protein. So one discovery leads to another. So now we understand that a lot of this, the way cells regulate growth is by picking up signals, activating a tyrosine kinase domain of the receptor to drive things in the cell. And we'll look at this in more detail in later slides, in later lectures. So we now know that many of these receptor molecules contain tyrosine kinase domains. When these proteins are mutated, which is often found in tumor cells, then the truncated version of the protein no longer regulates the tyrosine kinase, and the tyrosine kinase is overactive. So when you look at these different types of tumors now, and you look for mutations in these receptors that have tyrosine kinase domains, then there's a whole bunch of these different receptors that are mutated and are overactive in the tumors. And all of these receptors are tyrosine kinase receptors. And this was partly the discovery of these was partly driven by the understanding of what happens in the Src protein. So typically these receptors pick up signals from other cells and that turns them on to grow, but tumor cells often drive their own growth. They set up these autocrine signaling loops. So the signaling molecule is released by the same cell and then picked up by the same cell to drive growth. And that's often found for as a characteristic of these tumor cells, is that they have autocrine signaling, which is in normal cells this doesn't occur. So the molecular biologists now are starting to piece together the fact that they have these different oncoproteins. They've got a RAS protein, they've got a Src protein, they've got a receptor protein. And now they're starting to identify interaction between these different proteins in these signaling pathways, and they're starting to identify signaling pathways that control cell growth by identifying these mutated proteins. So effectively then, people had a good inkling, a good idea that these growth factors, these tyrosine kinases, they get overactive, and somehow they're signaling down to these other proteins such as the RAS protein. And then the combination of these proteins working together in pathways are driving cell growth. So we're going to look at the RAS protein in a bit more detail. So RAS has these lipid moieties attached to it, and because RAS is now attached to these lipid molecules, the lipid molecules embed themselves in the cytoplasmic membrane. So RAS is located on the plasma membrane, on the inside of the plasma membrane. And RAS itself tends to act as a switch. So RAS is in close proximity to these receptor molecules, and RAS is on the inside of the plasma membrane. And the receptor molecules cross the plasma membrane to pick up signals from outside, and maybe, who knows, maybe they transfer it to RAS. That was the idea. So let's have a look at what RAS is and how RAS turns itself on and off. So we know that mutated RAS on residue on codon 12 or on amino acid number 12, when it's mutated, it causes it to be active. So people have studied RAS and identified RAS in two states, an active state and an inactive state. So in tumor cells, RAS is locked into this active state and driving cell growth. So what's happening here? RAS is a protein, it's referred to as a GTP sort of A's, a GTPase. So it's a protein that has this small nucleotide as part of its, it binds to this nucleotide. And when it's bound to the nucleotide as a diphosphate, so a guanosine diphosphate, so GDP, when RAS is bound to the diphosphate nucleotide, it's inactive. Somehow in the cell, this RAS can also bind to a triphosphate, a guanosine triphosphate, and then when it's bound to the triphosphate, it's active. Okay, so you're all familiar with ATP being an energy molecule. Well, GTP is a molecule that regulates this RAS protein. Okay, so when RAS is bound to GDP, it's inactive. When RAS is bound to GTP, it's active. So the question is, how does the GTP switch between GDP? Okay, so we have two independent processes. We have a process of activation where GDP is exchanged for GTP. So one molecule of GTP is removed and it's replaced by GTP. And when you remove the GDP and replace it with GTP, you get active RAS. Okay, so there's a process of activation. There's a process of inactivation, and it's not through exchange, it's through dephosphorylation of that GTP to GDP. So effectively, that same nucleotide simply loses a phosphate group, and that causes inactivation. So we have nucleotide exchange for activation, and we have loss of the terminal phosphate group of GTP for inactivation. So effectively, we have hydrolysis of GTP to GDP. So there are clearly other proteins that work with RAS during the activation process, and there are other proteins that work with RAS during the inactivation. So activation is driven by a guanine exchange factor, okay, and that causes the exchange of GDP, and it's replaced by GTP. Inactivation of RAS is driven by hydrolysis of the GTP, and we have these other enzymes, these GTP activator proteins, and these activator proteins cause hydrolysis. Now, RAS has an enzyme activity, and RAS's enzyme activity is to hydrolyze GTP. So RAS has a mechanism to hydrolyze GTP to GDP, and when RAS hydrolyzes this nucleotide, it inactivates itself. Okay, it's a very weak activity, and these activator proteins make that hydrolysis activity much more efficient. Okay, so this protein allows RAS to hydrolyze GTP. This protein allows exchange of the GDP to GTP. Okay, so we have a process of activation and inactivation helped by these other proteins. So this is a summary of what I've just been saying to you. RAS was found bound to GDP when it's inactive. It jettisons its GDP, which is then replaced by a GTP, and then that turns RAS to become active. And then after a short time being active, the RAS is able to hydrolyze GTP back to GDP, and then it becomes inactive again. So you have this cycle of activation and inactivation. So you can think of it as a switch. You're switching RAS on, you're switching RAS off, you're switching RAS on, you're switching RAS off through this nucleotide exchange and hydrolysis. Okay? And when RAS loses its ability to switch off, and it's locked in the on step, in the on stage, and that constantly drives cell growth when cell growth should be off. Okay, so when you mutate RAS through that mutation on codon 12, it can't hydrolyze the GTP. So in context of cell signaling now, we have growth factors that bind to receptors. The receptors are somehow activating the RAS protein. So somehow these receptors are causing nucleotide exchange to activate RAS. Then RAS is active for a while, turns on these other cellular pathways, and after a period of time, RAS turns itself off. So the question was, when people didn't really understand what was happening, they knew growth factors were involved in cell signaling, they knew RAS was involved in cell signaling. One of the early hypotheses was that RAS was maybe driving a process to make a growth factor, and then the growth factor was being released and being picked up by the receptor, and that was driving things. So that was one of the early hypotheses. Turns out that was wrong. Okay, so turns out RAS is not upstream of the receptor. RAS is actually downstream. So RAS activation occurs after the growth signal here. So effectively, you've got a system in cells where you get a growth factor activating a receptor. The tyrosine kinase domain of this receptor becomes active, and through a series of steps that we'll look at in a later lecture, this leads to the activation of RAS, which then leads to the activation of processes such as cell growth or cell differentiation or other things. So we know that RAS can act as an oncoprotein, meaning when it's mutated, it can be a protein that drives oncogenesis, a protein that drives tumor formation. We've talked about the 12th codon or the 12th amino acid residue and some other codons that, when perturbed, cause this oncogenic activation of RAS. And let's have a look at how that mutation affects RAS. So we'll look at the 3D structure of RAS. So, I guess the question is, how can a mutation lead to the activation of an oncogene? So most alterations to RAS, such as deletions and things, actually inactivate RAS, okay? And they eliminate RAS from the cell, so that cell doesn't outcompete and outgrow other cells, and it's not an issue, okay? So the vast majority of proteins that strike RAS will cause it to lose its growth stimulation. But there's a couple of key mutations that can occur, and these drive it to become a hyperactive protein. So here's one of these ribbon diagrams that you should be familiar with, showing alpha helices and beta sheet regions and flexible loops within a protein. So all of these red and green and loopy sections are part of the RAS protein. And what's shown here in a ball and stick diagram, distinct to the protein, is the GTP or the GDP. It's showing the nucleotide that's bound to the protein. Okay? And if you look at where that nucleotide is bound to the protein, you're looking at this loop here. Somewhere along this loop is Gly12. It's the 12th residue. And we know that a mutation at that part of the protein is oncogenic. And if you look at where that protein is relative to this nucleotide, the side chain is really close to the gamma phosphate of GTP. And we know that GTP, which is or GDP, which is this region of the protein here, we know that the change in the GTP, GDP really affects the protein structure. And the ability of RAS to hydrolyze this gamma phosphate, the catalytic activity is the glycine 12 is part of that catalytic domain. Okay, as is the residue at position 61. Okay, so these two residues come into close proximity within the tertiary structure and the part of the catalytic domain that hydrolyzes the GTP. And when you hydrolyze GTP, the active RAS becomes inactive. So mutations at these key points stop RAS from inactivating itself. So again, earlier in the lecture I showed you that we had a point mutation at the 12th residue that leads to the formation of this oncogene, so that this residue, this glycine residue that we've been talking about, becomes mutated. So that point mutation now we're looking at in the context of the 3D structure. So that point mutation occurs at residue 12, and then when you stick a valine in there rather than a glycine, it loses the ability to hydrolyze that GTP. And effectively then you have a situation where RAS is on all the time because it cannot hydrolyze this nucleotide that's embedded within the protein. RAS also has these loops, these flexible loops, and depending on which nucleotide is attached, it changes the structure of this loop. So the gamma phosphate causes this loop to change. And I've got a little animation here that shows the the GTP or the GDP in the RAS structure. And this is showing RAS in the on state. And then if we run the animation here, you'll see that when RAS is in the... So see how this big loop changed its structure? So the presence of the gamma phosphate here. So when you switch from the GDP to the GTP, you cause a change in this part of the protein molecule. And as you get a change from the active GTP to the inactive GDP version, you get a change in structure. Okay? And when you get a mutation that stops the ability to hydrolyze that GTP, then you lock the protein in the on state. Okay, so hopefully looking at RAS in a bit of detail shows you how a single point mutation can change the structure of the overall protein, and it can lock the protein in the on step, on the on stage, and we'll look later on how an active RAS can work within a pathway to drive cellular processes. Okay, so thank you for your time, and we'll talk again soon.